everyone. If I say it 3 times, it's true. One of the great things about American University is we're a very collegial bunch. And I am sorry to interrupt your collegiality, but I promise you there'll be more opportunity for it. I'm Naomi Barron, Executive Director of the Center for Teaching, Research, and Learning, and I'm absolutely delighted to welcome you here for the second day, but this one comes with lunch, of our Teaching, Research, and Technology workshops that we hold each August to get you primed, ready, and excited about some of the new things you didn't know you could do a week from now, a week and a half from now when classes start. So we're delighted that you're here. We're using the luncheon today. Oh, first just a little bit about these workshops. Thus far, because we know people continue to sign up, thus far 195 people have said, this is a great way to round out my summer. So they've signed up for workshops and lunches and so forth. If you know other colleagues who still want to come and they didn't manage to do so yet, we have another full day of workshops next week, and you all have the schedule for those. What we try to do on at least one of the luncheon occasions in our August workshops is honor some of the people who have received awards from the Center for Teaching, Research, and Learning. Today we have 3 honorees, but they get to sing for their lunch, which I hope they've had a chance to at least start on. 2 of the awards that we give are for teaching in particular capacities. One is teaching with research, and the other is teaching with technology. Now, we would teach with all kinds of tools, and various people on the staff over the years have said, well, we should have a teaching with, and we go on and on and on, and then we would have lunches every day of the year to honor everyone who does this so well. But today we are honoring 2 people who have been awarded this year's Jack Child Teaching with Technology Award. For those of you who didn't know Jack Child, he was the founding director of the predecessor of CTRL, which is the Center for Teaching Excellence. And he passed away a couple of years ago, and we named the Teaching with Technology Award in his honor because he was one of the first people on this campus to seriously help faculty, encourage faculty to teach with technology. Do you want to hear it was using slides? That was novel at the time. Okay, goes way back. But the award honors faculty who have demonstrated creativity in using technology in their teaching. The 2015 award was split between 2 faculty members sitting here, John Brocht from the Department of Biology in the College of Arts and Sciences, and Sonia Greer, who I think is buried behind John, at least for me, from the Department of Marketing in the Kogod School of Business. And let me just mention what the next award is, so that Maria de Jesus doesn't feel, what am I doing sitting here? The second award is for teaching with research, which recognizes faculty who creatively incorporate original student research activities into their course experience. And the winner is, amazingly enough, Professor Maria de Jesus. But I'll tell you a little bit about her project in a moment. First, let me introduce Professor Brock, who's recognized for his, you can stand, you can stretch, we are informal around here, for his innovative and sophisticated use of advanced technologies in the classroom while teaching genomics, using a flipped classroom model. And if you don't know what that is, maybe you'll tell them. Professor Brock's students spend the in-class time to work with 10 different software packages while using AU's high-performance computing cluster, you'll hear what that is if you don't know as well, to assemble complete genome sequences. I'll stop at this point and welcome Professor Brock to tell us what he does with technology in the classroom.
Thank you, Naomi, for the warm welcome. Can you all hear me all right? Okay, so I guess I need this one. Just thought this might feed back. Is that okay? Can you hear me now? Okay. Closer, like that. Gotcha. So I'm really honored to be here and to tell you guys about the research and teaching that I did in the spring semester of 2015. In fact, it's listed right here. I've titled my talk Teaching with Technology in the Era of Big Data because we hear these terms a lot, big data, but I do genomics, and so I'm going to talk to you about what I do and how I integrated that really into the classroom. This was a graduate and senior level class, 596. And sort of the first time it was offered was the spring of 2015. Before I get into the details of how this class works and what a flipped classroom is, I have a question for you, for everybody, and you really don't need this, but what is a genome? All right, so we're teaching genomics. So if you were on the street and someone came up to you and said, what is your genome? Your answer would be, hopefully, all your DNA. Now, this is a little simplistic. All your DNA, what does that mean? Well, in general, people think of it as all 3,200,000,000 base pairs of sequence information in every single cell of your body. Now, in general, a human body has several trillion cells, so you could say all of your DNA is all those copies of the genome, but really you usually think of it as one copy of all of that DNA. Now, the reason why I taught this course and why this is an interesting field right now is because of this. So the cost of sequencing has recently taken a nosedive. So you can see, starting in 2001, when the human genome was finished, they've got the cost per genome at $100 million. Actually, the human genome cost almost $3 billion at that time, and that's when it was completed. And since that time, the cost of sequencing has dropped, sort of following what's considered to be Moore's law, which is the doubling of computer power every 2 years, processor speeds. So that's this line here. But sequencing, around 2007, there was a technological breakthrough. And the cost of sequencing just took this exponential plunge. And so by 2011, the cost of sequencing something like the human genome is down below $10,000. And this has been just, describing it as a revolution is sort of an understatement. The technology now allows us to see everything that's in your genome, not just a few genes, and it's just been fantastic. So some of the applications of sequencing are to sequence cancer, right? Sequence a tumor, sequence the normal tissue, figure out what's causing the cancer, and that's just exploded in recent years. Sequencing Alzheimer's or other diseases and trying to figure out what's driving, what's causing. Autism is another example, which I didn't put up there. People study the human population, migrations, historical patterns. It's a booming field. New organisms. One of the things I did in my postdoc was to sequence some weird organisms that do weird things with their genomes. And so this is a big area. Viruses. You can suddenly sequence lots and lots of viruses really cheaply because they're tiny genomes. Metagenomics, where you sequence all the genomes in a given environment, see what's there. It's just kind of exploded with the creativity of scientists. Obama just this year has released his precision medicine initiative with the goal of actually sequencing close to 1 million people. Now, not everyone's going to be sequenced, but a fair number of them probably will be. We currently have 10,000 human genomes on the books, and so this is just a field that's taking off. One of the things about genomics is that it requires good math skills because it's a lot of statistics and a lot of inference, and good computer skills and sort of a biological intuition about this data. So the data doesn't just talk to us. We have to actually extract answers, which means we have to know which questions to ask and how to ask them. And this combination of skills is actually fairly hard to come by. And this kind of sent, I think, the biological world reeling a little bit because we have just not been trained to do this sort of thing very well. So to put this in another way, this is the real reason why we did this course. So you have the evil sequencer in the background, right? And it's just spewing out so much data that this poor scientist is actually being strangled by the massive pile of information, right? And this really does sort of capture the state of biological research right now. The technology has just taken off, but really the ability to process the data and figure out what it all means is really lagging behind. And what makes this hard for us is that the sequence data comes out of the machine in millions of short pieces. It doesn't come out as a string of letters as it would be in your DNA, okay? So this is the fundamental challenge that we have to deal with. So I like this cartoon quite a bit. I think it might be hard to see, but this scientist has a puzzle that says 3 billion pieces genome, and there's this pile of pieces, and this guy says, well, I think I found a corner piece. And that's kind of where we are at this point, okay? So putting these pieces together is the challenge that this class was meant to address, and it takes enormous computational resources to be able to do that. Now, Naomi mentioned that we used Zorro. So Zorro is an AU cluster, high performance computing cluster, which we used. It has 15 compute nodes, and it performed great. So the way this works, when you do sequencing, you take genomic DNA, which is this squiggle at the top, and you break it into pieces, and the sequencing is to sequence all these little pieces, right? So you get all these little bits of data coming out. And I said here that it's redundant. That's because you actually sequence every piece of DNA multiple times. That enables you to go then and build back up the original sequence through assembly. So we did genome assembly in this class, where the goal is to take the overlaps between the pieces and reconstruct the original piece of DNA. Right? And so like I said, you start with millions of these little pieces, and you have to do all this reconstruction to build the original DNA. And it's actually far more complicated than you might think. It's not as simple as you'd hope, because the overlaps are not necessarily unique when you have that many pieces of DNA, right? So you have to deal with that. There are errors that accumulate when you sequence, so how do you deal with mistakes? And that messes up your assembly. It's really complicated. And it doesn't give you just genomic DNA sequence. It gives you lots of pieces of DNA that sometimes you have to assemble further. But it's a start, and it's pretty powerful. So for this course, the questions we wanted to address, and these are really pretty important questions. Once you have genome sequence, what do we do with it, right? It doesn't just, we can't just read it like a book. There's still meaning we have to extract. For example, is it correct? And how do we know? And this is one of the big questions, I think, for genomics. How do you know what the right answer is when you don't have the right answer to start with? So there are some ways computationally to get those, to answer those questions. What does the genome say? What genes are there? What repetitive sequences are there? How does it compare to other organisms? There's all these interesting questions. But ultimately, what is the biological meaning of the data? And I wanted to instill in students, this is what we want. We want to get the meaning out of the data. So the course was designed as a flip design. So in class was reserved for students doing computational exercises. Now this wasn't always, a lot of times they would kick off a program and let it run for a few days. So they would kick something off, and then by the next class session we could evaluate the results. But teaching students how to start things on Zorro was something that would take a few minutes to introduce. So I always had an intro lecture to orient students. We had great help from the CTRL and Zorro staff on how to run these programs, so big shout out to them. And we actually processed real experimental data from my research in this course. So that was really, it gave an air of fun because we're doing some new things that have never been done before, and we're seeing some things for the first time. So the students really got into it. There were some outside readings, research articles, papers from the literature. Often I would assign the paper that describes the program that they were going to run that day. So they'd at least come in with some basis of knowing what they're dealing with. There was a textbook, but it was only used slightly, sort of as background for the students. Students gave presentations in the course. We had reserved presentation times for cool genomics, where they could find the coolest paper they could find and were most excited about and talk about it. And those went great. Students got very into that. And then a final research presentation. So the idea was to, at the end of the course, put all these pieces together in a novel way and address a real question. The students did a great job. Some of those presentations were fundable proposals. And a daily quiz, just to encourage students to read the paper of the day and take attendance. So the very first course lecture, this was, I think, my first slide in the class. And this is the entire course in one slide. So really, it's a flow chart from raw data up here, which I gave the students, genome assembly, genome validation, genome annotation, and genome comparison. And that's sort of what I just described to you in words. So putting the genome together is on Zorro. The rest of this, validating, annotating, and comparing it, these are all programs we ran in a virtual computing environment. So these programs actually don't run, most of them don't run on Windows. And we were using Dell workstations in the CTRL lab for this class. So we had to improvise a bit, and it turns out that you can actually run Linux on any workstation that you want. And what you use is something like this. This is called VirtualBox, and it's a virtual machine that can run any platform that you want. So actually you could run, this is a Mac, you can run Windows on the Mac, you can run Mac operating system on Windows, you can run Linux on either one, or vice versa. So here I have a Linux machine. Actually, this is the Linux virtual machine from the class. So I'm going to start it right now. This is the console that you're seeing. The students would come in and click this, and it takes a moment, but it's now starting Linux. So now Linux is running on my Mac. And now this is a Linux machine running on the Mac, which is kind of neat. And actually what I did is I preloaded all of the software for the class onto this virtual machine. And this was installed in the CTRL lab, thanks to a lot of help from Eric Charles, who installed this. But this was great because it's one package that has everything that they're going to need. So every time they came into class, they could start this machine and run their computations. So I actually want to show you guys, don't worry, I'm almost done. Show you guys what some of the data looks like, and then we'll wrap this up. So they would come in, and this was the terminal where students would actually run things. And hang on just a second, I need my other hand. Then we would go. So here we can actually see my presentation on my desktop. We can see the data, and what we can do okay, that's some actual data straight out of a sequencing machine. And you can see it's actually strings of A, G, C, and T, but they're short, right? So they start over here, and the string goes and it wraps. This is information about the quality of this string here, right? So it's very cool. So you actually get a lot of information. Now this file, I'm now scrolling down, you can do this for days, and you might get through, you know, a fraction. Maybe you'd get through a tenth of this file in a week. I mean, this file is huge. You can never look at your data. That's one of the weird things about genomics. We can never just sit and look at the data. You have to query it in some way. And so the goal was to teach students how to query it. Okay, I won't make everyone dizzy. When they were done at the end of the class, you just go here and you power off the machine, and it shuts down, and there you go. And the nice thing about this is you can export this machine onto a flash drive and give it to students. They can upload it onto whatever computer they have at home and take it with them, right? So it becomes sort of a keepsake, I told them, but also it's powerful. So if you're doing genomics, you have everything installed. You don't have to spend time installing the software. It's ready to go. So a number of students actually took me up on that and have used it. And some of the students are still doing genomics with me, and they're using that particular VirtualBox machine. So lessons I learned. The students learned a lot. I learned a lot in this class. Technology has a lot of challenges. I spent a lot of time optimizing how we're going to do this in the class because our time was limited. But what's cool about running programs is there's a sort of feedback loop, right? So I teach them a concept, then they start their Linux, and we type in a command, and you get some results sort of immediately, or relatively immediately, maybe several minutes later. And then you evaluate that result. Maybe the machine tells you, oh, you didn't type it right, or I can't find a file. So it worked out. It's a new way of learning. It sort of feeds back to the student immediately about their understanding. Troubleshooting is definitely a skill the students learned, and I tried to encourage them to learn. Balancing the progress of a whole class doing this with the progress of a few students who might be ahead or behind. That was an interesting learning process. And there's some research insights from running this same assembly with different parameters. And each student had their own parameters, and they signed up, and then they analyzed the results. And so that was actually pretty useful. Like I said, they could take this with them. So that's basically it. These are some of the programs students ran. We won't spend time on it, but these are some of the commands that they entered. And really, I just want to acknowledge that there were 13 students who were brave enough to take this course. 10 of them masters and 3 undergrads. They all did great. I really appreciate all the support from the Zorro support staff, especially Asen, Angel, and Evgeny, and the CTRL lab and staff, especially Eric, who helped me a lot with this, and the entire CTRL staff has been great. So thank you.
I think we'll wait until all 3 of our colleagues have had a chance to tell us about their work before we take questions. I'm also stalling slightly for time while we change computers. What I can do productively as I stall for time is to say, I'm delighted you came to AU when you did, because had you come 5 or 6 years earlier, we wouldn't have had this capacity. And where is Asen? Asen, I saw you. There you are. And a big shout out to Asen, who really spearheaded our getting high-performance computing going, along with our colleague in economics who got the grant from the National Science Foundation that made it possible for us to get the project really launched. And Angel is here. Angel is not here. Angel may be, he's not here. Okay, so shout out to everybody, but Angel Bogashev is the person who is most hands-on responsible for doing high-performance computing, and Eric is sitting here, so thanks to everyone who made all your wonderful teaching with technology happen. Shifting gears, Professor Sonia Greer is recognized for her eclectic and innovative incorporation of technology in teaching. Most prominently, Professor Greer shoots, in the best sense of the term, and edits her own films, yes, films, about the topics she researches and teaches, making topics more accessible and engaging by adding an audiovisual element to traditional course readings. Congratulations.
Thank you. And thank you all for being here today. Now for something completely different, from high tech to low tech. So I'm going to talk about the use of low tech, everyday type tools for high impact teaching. First of all, when I got the email about this award, I was like, huh, who, me? That I'm really a low tech type of person. I don't use any social media, no Facebook in my class, no blogs, not even clickers. And personally, I still prefer email to texting. I struggled teaching undergrads when I came to AU 10 years ago, although I had great teaching ratings with MBAs at Stanford. So it was really, I spent my first few years really struggling, trying to find out how to resonate with the undergrads. And I utilized a lot of the teaching resources on campus. I've been to every Ann Farron, I've been to most of these teaching with technology workshops, and I'm always open and willing to try whatever might work. And what I really gained from a lot of those workshops and resources is that students want accessibility. They want accessibility to class materials. They want accessibility to the professor. They also want engaged. They want to be engaged. I don't want to say entertained, although people say that maybe that's what they really want, but they really want to be engaged with the class material and to find it interesting from their perspective. And a lot of this comes from being a marketing professor. We always think about targeting. What does the target market want? What does the customer want? So it can't be from my view of what I want them to have. It really has to be me understanding what they want and need. So what I teach, I've taught 2 primary classes that I think are relevant here. One is advertising, which is really about developing promotional campaigns, ads, all kinds of integrated marketing, digital marketing, etc. The second one is a course that I created called Marketing for Social Change, which is about applying marketing to social challenges like drunk driving or texting while driving or drug use, etc. And both courses really focus on understanding consumers in order to develop marketing strategies to try to influence their attitudes, values, and behaviors. So this is what I want to convey to students. So my approach to technology is really, I want to fulfill what it is that they want this access and engagement. But I also want it to have a significant impact on their learning. But I do have a concern about the technology overwhelming the learning, about having too many bells and whistles or something that might actually overwhelm me. I also need it to fit with what my beliefs and preferences, both personally and professional, are, because if it doesn't, I'm not going to do it on a consistent basis, or I'm not going to be effective with it. I need it to be easy to learn, easy to use, and not a lot of setup time. So I'm really looking for the sweet spot, this low tech that has some high impact. The way I use technology, the goal of my use of it is really to deepen their learning by supporting these instructional objectives. And so I think about it in two different ways. One is as a marketing channel. So the channel is what connects the product to the consumer from the manufacturer or the distributor. So what I'm trying to do is connect this material to the students, and this technology is my channel, much as we'd see TV is the channel that you put an ad on. I also think about it in terms of transmedia storytelling, and this is something that's evolved a lot in communications and marketing over time, but it's about telling the curriculum in the way of a story, but telling the story in bits and pieces in different media, because students will learn different things from different media and they'll remember different things, and so that's a way of engaging them. So from a transmedia perspective, I really focus on the content of the course and the context of the types of problems and issues, whether it's promoting a neighborhood or promoting a particular product, or whether it's trying to get teenagers to not text and drive. You really have to understand the context of those problems, and so I try to use technology in that way to show the behavior, because I could say, no, texting and driving is bad, and you should tell kids that, but when you see that kids are on their way somewhere and they're trying to do it and they actually visualize the problem, they have a better understanding of the challenge of developing a strategy to try and change it. So I use all of these different kinds of low-tech things like Google Doc, Panopto, which will soon be Kaltura, I guess, multimedia resources, film, photos, Skype, and PowerPoint, and I'm just going to give a couple examples of these. So one is for class logistics, dealing with the accessibility piece. I use a Google Doc to set up the required meetings that the students have with me. They have to meet with me at least once every semester, and I took that directly from one of the Ann Farron sessions with Chris Palmer. And it really helped me to understand who the students were, and for them to see that I was open and I was there and they could come and talk to me at any time. And it's made a huge difference in my class. But instead of having them come and sign up on the class or sit and wait in a group outside during office hours, they can set up and schedule their time with me over the web. And they can change it, and so there's this real-time sense of meetings, and you wouldn't believe how many students mention that as something they really like about the class. Skype, I bring experts into class, and I'll talk a little bit about that a little later, and then a YouTube playlist. And so I'm able to pull up everything from ads to TED Talks to company profiles, etc. And I have a playlist for each class, just like this. This one is for the social and legal class in my 411. And so everything is there. It limits any kind of in-class setup, and you can easily change it, remove, or even add during the class to this. Show and tell is something else, another assignment that the students have that I've done since I've been teaching, and where students come to class and they have to bring something, everyone signs up, that relates to the day's topic. And so this allows them to bring in something that interests them. And it might be internet, it might be something YouTube, it could be from social media, it could be a picture they've taken on the way to class, it could be some new technology that they talk about, and they have to relate it to the day's class. And then they also have to have a question for the other students. And so this helps to build students thinking on their feet. It brings in new materials. It increases this engagement and exposure, their facility with applying the different concepts. It's also my research. I get to see what they're interested in, what they're thinking about, how they're thinking about the course, and it also gives me updated materials that go right into my YouTube playlist for show and tell, and then I can pick the ones that I want to use for different courses, different class sessions. And then the last example I'll use is film. And this is something that I started doing on my sabbatical. I made a film about Cuba, about capitalism and collective well-being in Cuba. And that was something that I used for a case that I had written several years ago on marketing Cuba to Americans. I met a photographer who had this idea about developing a photography exchange, a cultural exchange, because it was built on the notion that Americans don't know anything about Cuba, and one day it would open up, that day has come, and that all we think about are the stereotypes of cigars and old cars. And so I wrote this case about how would you do that as a matter of social change to try to educate and learn and teach Americans about Cuba. And so while I was there, I developed this film, Citizen Consumer. I then, the following year, I made another film called Dog Parks and Coffee Shops about gentrification in 3 Washington, D.C. areas. I had never made films before. I had no training other than over in the multimedia lab. I took the class on Final Cut Pro. And so both of these films now go with cases. I have a case on marketing pet worth that the class few years ago had worked with trying to market the business association in Petworth. Michael Mados is very helpful in coming to visit the class and showing them tools online that they might use to get information. We wrote a case following the students, working with them, and then I now use this film. Now the film can show them about gentrification, for example, which isn't something that's part of the marketing strategy per se, but if they're going to develop a marketing strategy in an area that's gentrifying, there are lots of issues that are going to relate to that they can see. It also will bring up issues like race and class, which might be more sensitive and harder for students to sort of talk about in general, but when they see it and can visualize it, it makes those conversations a lot easier. So in summary, I'd say there's many more flashy and elaborate tools that I could use, but these are free, easy, and familiar. And although I might use some of these in the future, I think that these low-tech tools can really increase the students' access to the class, to the materials and the professor, and enhance their learning. Thank you.
So yes, there is technology. But there are some other things as well for which we give another award. Although there is research that has gone into the first 2 presentations of Sonia and of John. The other award, which I mentioned at the beginning, is our Teaching with Research Award, where the research is not, so we as faculty are doing research, that's good, but rather how are we training our students to think about, to understand the parameters of, to engage in research themselves. And sometimes that does combine with our own personal research experiences. And this year, we recognize Professor Maria de Jesus for her creative efforts to, if I can read, integrate her research with her teaching by engaging students in all the steps of scientific research in all of her classes, and for mentoring students as they develop their own research projects. For example, as part of her recently funded research project on HIV and HIV prevention with African American and African immigrant women in DC, she designed assignments for her course in which her students analyzed a subset of her interview data with the aims of learning to analyze and interpret data to make valid claims. Maria.
Thank you so much, Naomi. Can you all hear me like this? Okay, yes? Okay, this one? Can you hear me better this way? Okay. Well, I can seriously spend hours talking about this, and I imagine that this is basically catering to an audience that understands the importance of research, but especially the engagement, as Sonia was talking about, the engagement of our students with our own research, and seeing themselves as researchers. And I think this is really important, both for our undergraduate and our graduate population. We assume that people know what it means to do research sometimes, and we assume that we understand what it means to think of ourselves in terms of a research identity. But many of our students, especially my understanding and my experiences here at AU, have taught me since 2010 when I first started, that many of our students don't necessarily relate. They can't see themselves as researchers. So part of the thinking and a lot of my inspiration actually came from CTRL and NFAIR conferences and workshops, really thinking about, well, how do I bring these 2 worlds together, the world of teaching and the world of research? And certainly this fits with the goals of AU, particularly the goals of becoming a student's, a research scholar, a teacher scholar, if you will. So just a little overview of why it would be important to think about collaborative research. And again, many of you are probably just as passionate about this as I am in terms of getting students involved and engaged. So a lot of us think about AU and coming to AU, both as faculty, staff, certainly students, to do something with your knowledge. So it's not just about gaining knowledge, it's about service. What do I do to change the world? How do I become an agent of change? A problem-oriented approach to teaching, which excited me. I remember teaching my first class, and my ratings, although good, could have been a lot better. And part of the reason for those ratings were because it was too theoretical. And so I learned right away that these students, for the most part, want practical experience as well. And so that's what really got me also to start thinking about, well, how do I get them engaged in my research and in their own research projects, if that's what they choose to do. A Freirean pedagogy, again, this idea of learning by doing, not simply learning through book knowledge, and helping students on the professional job market. So I now have a number of students who have been able to land jobs either through an internship process or directly because they came out with a research project, can talk about research in a way that they sound knowledgeable and experienced, and that allowed them to get their foot in the door into the professional market. What motivated me, and I think a lot of other faculty members, to involve students in research? Well, thinking about the complex questions of the day, which obviously takes an adoption of a multidisciplinary approach, requires a multidisciplinary team. So a lot of times people think, well, okay, well, I can see everything that students have to benefit through this process of collaborative research, but what do you gain as a faculty member? I gain a ton because, again, as my colleagues just talked about, fresh eyes, motivated students for the most part, who are willing to engage, wanting to engage. And so they begin to think about these questions in much different ways, begin to add input that I wasn't necessarily thinking about, hence, changing the way I'm starting to think about the actual research that I'm doing. Again, highly motivated students. For those of you teaching for the first time, I think a lot of our students are motivated, and I think it's the idea of stimulating them even further, and again, by becoming engaged with them. Building a cadre of researchers, future professionals, whether they're practitioners, teachers, scientists. We want them to do something with this knowledge and be able to value it. And then shape, again, those who will be the future professionals. A little spiel, again, on how students can help faculty with research, certainly through specific skills around grant support, grant making. And I'll give you an example that I used in both my undergraduate class, Health Across Borders with Immigrants and Refugees, and my graduate research class on global health, culture, and communication. All of them love to write a grant. It was a mock grant proposal as one of the assignments. Of course, I gave them an opportunity to see what a model of a good grant looks like and just not expect them to know that. What the criteria are from the perspective of an evaluator of that grant looks like, and then step by step guide them through that process. That was a learned skill, again, a research skill. It's not meant for every research class, but I used it in these two particular classes, and that was an assignment that they absolutely loved, which people thought, you're crazy, you're gonna get these students to write a grant. They loved it, and many of them actually turned it into a fundable research grant. So they took it and submitted it. Statistical help, again, a shout out to CTRL. I don't feel like I'm alone on an island. I feel like there's so much support out there, meaning also that my students have extra support. So both from me for statistical help, qualitative research, analysis help, but also leaning on CTRL to also be that beacon of guidance. Interpretation purposes, so to analyze data, and again, having a debriefing process, a group process, which allows the students to really think about what it means to interpret data. What is the story that your qualitative data is telling you? and gaining consensus around that. Again, reading research. So a lot of our students, I think, you know, we're giving them research papers all the time to read, but it's another thing to actually get them involved in the research process firsthand to gain that direct knowledge. In terms of field research, and I'll tell you a little bit in the next few minutes about an actual research project involving the HIV research that I did last year, more bodies can cover greater territory. So in this particular case, we were looking at the metropolitan Washington region. So if you know, it was nice to have student researchers, some of whom, not all, because not everyone wants to do field research, but for those who wanted to do this, this was an amazing way to gain research experience, doing focus groups, doing interview, and going out there into the field. So I could get more data, which then allowed them to be able to analyze as a group, and et cetera. And bring in, again, more diverse perspectives. So these were some of the other things they were involved in terms of literature reviews, grant writing I talked about already, statistical qualitative analysis and collecting data, co-writing papers. So many of them, not many of them, but many of them wrote papers, obviously, for the class, but some of those papers turned into published peer-reviewed articles in high-impact journals. So that was also something that they could put on their CV that they were proud of, they learned a ton through. So a lot of the project, the ideas and the experiences came out of the HIV research that was funded by the Developmental Center for AIDS Research. So I wasn't able to pay the students, but they were all able to gain some of these skills in one way or another. So part of it was figuring out, well, what are your talents, your research ideas? What are your research strengths? And all of them have these, even though they may not necessarily see themselves as researchers. I asked them to first go through a process whereby they could figure out if they were strong writers, if they were more technologically oriented, if they were more interested in developing graphs and visuals. Once I figured that out, I had little subsets, groups that worked together, so they weren't on their own. I think none of us work as researchers independently, so it was this idea of learning through doing, through a collaborative process, also with their peers and with me and other resources on campus, to be able to break apart the research process from beginning, as Naomi talked about, from beginning to the end. So developing a literature review, what do we know already? What's out there in terms of HIV and HIV prevention in the Washington metropolitan area, in particular with African immigrants and refugees? Very little. That's what we came up with. Very little was done. So that was a first step, gaining knowledge, gaining information, which meant we have to do interview data. We have to get focus group data. We have to learn from the ground, given that we don't have much data, what the experiences and the perspectives are. So this became a community-oriented, participatory, qualitative research project. Some of the students, a smaller subset, went out and got some data, some of which I already had, so they were able to integrate the data that I already had and analyze it together to then, after the analysis, to then write papers about it. And again, some of these papers were published. So very exciting. I think it was co-learning, both from a student perspective and a faculty perspective. Would I do it again? Yes, because I end up with published papers and I end up with motivated students who come back, many of whom are now part of a research team. So beyond the classroom, they were inspired to then come back and continue on in a different capacity with me. And some of whom decided to take research in a different direction and, you know, ended up doing independent projects with other faculty members, which is also very exciting. And most of all, I think, to begin a process whereby these students began to see themselves as potential researchers, whether they were consumers of research, whether they were producers of research, they began to identify with that. And to me, that was, I think, one of the best benefits as a faculty member, to see that students could sort of change their perspective just within one semester. So I'll end there, so we have a few minutes for Q&A for all of us. Thank you.
Thank you so much. no, CTRL did not solicit any unpaid, except for lunch, announcements about what we do. But it was a genuine pleasure to hear each one of our speakers talk about ways in which they have grown, they have changed in terms of their professional growth and their lives specifically as teachers. So bravo to all of you. Questions? Anna, you want to run the question? I think Betsy gets her hand up really fast. So Betsy goes first, and then after that, someone else gets to.
Kind of quick. It's for Maria. It sounds fabulous. I mean, I'm just sitting here thinking like, oh, I want to be your student. But as faculty members, I'm wondering what others might be thinking, which is, how much work is it? How much extra work is it for you to mentor and train these students to be researchers and to actually produce products that are publishable in a semester?
And you know, writing is about rewriting and rewriting, so it wasn't like their final paper was ready for publication. Let me make it clear. We had to go through many iterations before we got it there. When is teaching not work, right? So depending on how we view it, you know, teaching is always going to be work-heavy, time-consuming, and then inspiring. So for me, it was about, well, figuring out what I like and integrating it into my teaching, and then hoping that other people like it too, so that it becomes contagious. So a little bit, you know, it's a self-selected process as well, because these were elective classes, they weren't required classes, which I think makes a difference. And so they have a sense already, just even from reading the description of the class, that they are going to be involved, that it's high expectations. At the same time, you know, once I think we've taught these types of classes once or twice, you know, our names go around, the reputation goes around about the particular classes, and so they say, look, don't get intimidated. Like, she's good, take her, she's fine. You'll be able to do well in this class if you're motivated. And I always tell my students, you know, when they ask, what type of a professor are you? Will I get an A or a D? I said A through F. You know, it's really, what are you willing to give to yourself? So, and I sort of, you know, I have the little disclaimer that says, you put in 100%, I'll meet you 150% of the way. Like, I will go overboard, but I need to see you coming through. So it's a two-way process. It is a lot of work, but as I said, I also gain from it. It's not a one-sided thing, so I think it's worth it. I knew you would.
I have a related question. I have a related question for Maria, which is, how in the world did you manage this logistically? Because I think it's awesome, but I know how much time it takes to like apply for a grant through an organization. You get permission, you have to change the protocols, then get IRB, and I mean, is it a cart before the horse? So did you think of this like a year ahead of time, and then you just filled in, you know, the people who were going to do the interviews and focus groups at the last minute, depending on if the class worked out? How did you do that?
Well, this one was a little easier, and we could certainly get into the details later, Lauren, but it was already a funded project, and so I already had data to work with. I had pilot data that I could work with, but I wanted to give them a taste of, well, what is it like to do an interview, you know, and be trained in it, and what is it like to do a focus group? So I wanted them to also gather some data for those, you know, that small group that wanted to do that. But I already had some data that they could work with.
And was it a grant that you did that understood that students would be doing this as a part of it, or was it just an external?
No, I mean, I had written a piece in the grant proposal, you know, that there would be some student involvement research, but at the time, you know, the idea hadn't germined yet about, you know, I can actually do this for real. Like, I just sort of put it in because it sounded good. But then it actually happened that way. So the timing, you know, it was, you're right. I mean, there were certain things that were out of my control that just worked really well. I was lucky enough that the timeline worked. You know, the class was offered, there were enough students, so it wasn't canceled. I mean, there were just certain things that fell into place. But I think there are different models and there are different ways to work around that. If one doesn't have pilot data or doesn't have funded research, you can start from scratch in some ways and do something much smaller, a much smaller scope project. But let's talk details later.
Any more questions?
Sonia, I wanted to ask you, for the show and tell, how large was your class size to be able to do that?
This last class was 27 students, but I've done it every semester, and I've had classes everywhere from 38 students to 18 students.
So for the larger size classes, how did you make sure that everyone had a chance to?
We might have 2 or 3 students who go in a day, but everyone gets an opportunity to go. And it's become increasingly formalized over time. It used to be whoever wanted to come in, and it would add to their participation points. And now it's more formal where everyone is required to, and it's only worth maybe 3 or 4 points, but the students really get into it, and they always mention it as something they really like about the class.
John, can you mention one or two of the titles of things that got published or got submitted for publication?
You know, we didn't actually submit anything. It's just when I was reading their grant proposals, they were definitely some that were really innovative and really well thought through.
Can you say one or two? I like
one of them was looking at the nutri-epigenetic effects of switching to a vegetarian diet. So I thought that was really cool. So the idea was that when you go from eating meat to being vegetarian, which a lot of people do, what does that do to your genomics? And nobody's looking at this. It turns out there's a fish that does this naturally. I think it was a carp that does this during its growth. As a larva or young fish, it eats meat, and then it transitions to an all-plant diet. So she was proposing to study this process. I thought that was really innovative, and she was an undergraduate, so she submitted one of the better. But there was just a broad range. I had another proposal was on in India, a lot of people get treated for malaria, but they end up getting rheumatoid arthritis. So a lot of these drugs that inhibit the immune system will actually have these other side effects. And she was looking for genetic determinants of that and whether we can predict people who might be okay on the drugs and people who wouldn't be okay on the drugs. And I thought that was also really great. These sorts of ideas that we just don't always think about when we're enmeshed in our standard model systems. And so yeah, I thought it was really, really great. But just to clarify, none of them have been submitted yet, but I think that a number of them could be for sure. And these students are still here and they're sort of progressing through. So tried to encourage them. These proposals were 2 pages long, by the way. I told them everything needs to fit in 2 pages. Just makes everything easier. And that's the way people are moving these days, too.
Maria, you mentioned with your students doing research on a project you're running. I'm wondering, is there any issue about paying the students that could come up? Happen to mention, I work for the Department of Labor, and and I don't investigate, so don't worry about it. But it comes across my desk all the time, issues not so much dissimilar from what you're saying. And I don't know if you got advice from the administration or what, but you got to be careful sometimes about that.
Yeah, I mean, certainly ethics comes first, both, human participants for research projects and certainly our students. So, we had a long talk about this in one of our School of International Service, SIS workshops as well, and this came up as well, not only in the context of a class and bringing students in to learn about research and do research, but also in the context of, engaging with a research assistant, for example, and making sure everything is clear in terms of what their tasks are and what they're getting paid for, et cetera. Yeah, I mean, this is something that I think is important to think about. I think in my case, they had very specific tasks that they worked on. Everything was sort of a selected process for them. they had the option of doing field research versus a literature review group versus statistical analysis versus, and by no means would I take anything that they did and publish it on my own. So any of those who were interested, actually my goal wasn't necessarily to, publish anything. This was coming from the students afterwards. A few students were self-motivated to then write something. But I hear your point. It's a fuzzy line between sort of the research ethics and, the fact that they're students and they're not being paid. So making sure that they're not being used in an exploitative way or as cheap labor to get your projects done. I remember going through that as a graduate student myself, where people did take what I did and publish it and didn't think of crediting me in any way. So, yeah.
It has nothing to do with ethics. It's the law. I mean, I'm not asking about ethics. I'm just wondering if you're violating the law or not. I don't know, but it's something you might want to find out.
Let me weigh in on that for just a moment, if I might.
Sure.
As many of you know, AU, particularly for undergraduates, although for graduate students it's been the case for a long time, AU is looking to enhance opportunities for undergraduates to begin thinking, to begin developing researcher minds and to start doing research from their first year here in some cases. So the question of what role students have in projects that are crafted around something a faculty member is working on already is going to be a larger issue institutionally. So it's not entirely on your shoulders, it's one that we have to think about institutionally. What is clear from my own experience, because sometimes it has been students working, graduate students working for a salary and sometimes undergraduates as part of a class project, is if something is indeed part of what students are getting academic credit for and learning from, and I would absolutely say getting credited for if something does end up being published. I think there are ways that we as a university, plus many other universities that look to enhance, particularly undergraduate student research, can find really good win-win situations. I can promise you, mentioning students' names in any publication to thank them for their efforts is worth much more than money to most students. That doesn't mean we shouldn't pay them if it's appropriate to do so, but there are ways to acknowledge what students do and how they contribute to our class. So I'm thinking about Sonia's experience where students are bringing in things for show and tell, and then that becomes part of a class structure because it was really good, and Sonia may not have known about that particular one. Or we'll see what direction John's research is going to go, and maybe you really want to know what it means to go vegetarian. Look at widows in India who go vegetarian. You've got a whole big population, to, in addition to the fish, are some Americans to look at. Let me stop. Right, and that's the internship kind of a rule, but we need to think about this as well for research. You're entirely right. Let me stop at this point. Before I thank the speakers, because otherwise you'll get up and leave, let me just mention a couple of things. Your tables are covered with all kinds of nice flyers. I'm not going to go through each one, except to say there are lots of other things we do in CTRL. There are programs we run in the various aspects of who we are. I don't think you have flyers on your table about two other opportunities. If they're there and you say, it's on my table, please tell me. One is we have what are called teaching enhancement grants of up to $100 to help do something in your course coming this fall that otherwise you might not have been able to do. Maybe bringing a guest speaker, maybe helping defer cost of admission somewhere. One of my favorite ones was a couple of years ago, we had somebody teaching theater. They were teaching Cyrano de Bergerac, of the nose. And he wanted noses for everyone to wear as they were playing the roles. Okay, so however creative you can be, we funded it. It was great. The project worked beautifully. We did a Japanese tea ceremony. We've done many, many things. All you have to do is go onto our site, go to the CTRL site, go to awards and grants, and you'll find where to apply. The other opportunity coming up is for open educational resources. Lindsay, did we get a flyer out or no? You don't have them, so we probably don't. Go online for awards and grants, and you'll see the ways in which we are helping support faculty develop recastings of or brand new courses that make heavy use of what are known as open educational resources, and for the oversimplified definition, and Lindsay Murphy here is spearheading the project, it's a way that students can get really good educational materials that do not have costs attached to them. And we have funded a couple of faculty members to start this enterprise. In fact, you're doing a session after lunch. In a few minutes, you could come learn more about it. But if you can't stay for this or you're going to another session, you can learn how we can help support your activities in fostering broader educational opportunities, as well as ways of not having exceedingly expensive textbooks that your students may not buy or read. That said, let me have one more announcement. There is an evaluation form, and there's a box that Anna Olson is holding up. Please fill it out. Tell us what you like, what you don't like about lunch. No comments on the food, because we cannot fit everybody's tastes. Just let me know if you have a problem. But don't. Then just to say here or something. Thank you very much. So let me just thank our speakers, to thank Maria, to thank Sonia, to thank John. And I'll also just take this opportunity to thank Anna Olson and everyone here from CTRL who has in one capacity or another helped make these 3 days or 4 days of workshops. Well, really 4 days. Three and some pieces happen. Have some more food. There's still some left, and we hope to see you at whichever of the next afternoon sessions is right for you. Thank you.